Okay, so you are studying cell biology and you have come across centrioles and cilia. They sound so similar, right? They even look alike under a microscope. But here is the kicker. They are nothing alike in function. If you are confused, you are not alone. I bet you have been wondering, why do they have the same structure? How they are connected to each other? And why we even need them anyway? Today, let's break down the confusion and make it simple for you. In this video from Genaura Academy, we will explore the fascinating world of centrioles and cilia. Two tiny yet crucial structures found in the eukaryotic cells. Yes, both these structures are found in the eukaryotic cells. So today we will learn about their structure, function, role in cell division and movement, all explained in a fun and engaging way with simple language and real life comparisons. Let's talk about why these two are so confusing. So both the centriole and cilia are made up of microtubules. They look alike under the microscope and they both seem to be involved in the movement in same way. But here is where things go tricky. Centrioles are static structures. They don't move themselves but play a role in organizing movement like during cell division. Cilia, on the other hand, move. They are tiny oars pushing things around or sweeping stuff away. So why do they look similar but their roles are so different? Let's untangle that. Imagine this. You are at a train station. The centrioles are like the track switchers. Centrioles act as the track switchers. They don't move, but they decide how and where things divide and organize. And the cilia are the tiny moving bushes. Tiny moving brushes on a cleaning train. They actively sweep and move stuff around. Now you get the idea. One is the planner and the another one is a mover. So you can say that centriole is a planner and cilia is a mover. Now let's first talk about the centrioles. We can also say them as the organizers. Centrioles are organizers. So centrioles are the cylindrical structure made up of 9 triplets of microtubules. 9 triplets of microtubules they are made up from, and they are usually found near a nucleus in the cell and comes in pair with one perpendicular to the another. Say for an example, this is so always one perpendicular to another. Here is the perpendicular direction. So this is how they usually found near the nucleus and come in pairs with one perpendicular to the other. So you might wonder that what they actually do. What is the key function of them? So centrioles are the masterminds behind the cell division. Mastermind behind the cell division. They help organize the spindle fibers that pull the chromosomes apart during the mitosis. Now the question is, do they move? Not really. They stay in one place and their main job is organizing other structures for the movement. They are acting like the conductor of an orchestra. They don't play the instruments, but they make sure everything happens at the right time. Now the confusing fact alert. Centrioles are also the basal bodies from which a cilia grow. That's why they seem related. Now, let's talk about the cilia. We can also call them as the movers. So, cilia are the tiny hair-like projections that stick out from the cell surface. They made up of a special structure, and that structure is known as axoneme, which follows 9+2 arrangement. For this centriole, they were following 9+0 arrangement, and the cilia is following the 9+2 arrangement of microtubules. So what are their job? What are their key functions? So cilia do move. They use a rhythmic back and forth motion to either move substances like mucus across the surface of the cell or help with the cell movement. So there is a question that do they originate from the centrioles? Yes. Cilia originate from the centrioles only. Cilia grow out of the centrioles, and the centrioles are called as the basal bodies for the cilia. Now the question is, do cilia divide cells? No. Unlike centrioles, cilia are involved in the active movement but not in the cell division. Have you ever looked at the diagrams of centrioles and cilia and thought, wait, they are not, they basically the same thing? Both are made up of microtubules, but there is a difference between their symmetry. That's why they are doing totally different jobs. So at first have a look on the cilia. This outer membrane is the cell membrane, and inside this the microtubules are arranged in a doublet form, and the doublets of the microtubules are linked together by the interdoublet bridge. The linkage between these doublets of microtubules is known as interdoublet bridge. In between, and how many microtubules doublet are there? 9 microtubules. Now you can see 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 microtubule doublets are there, and these doublets are linked together with the interdoublet bridge. And in between these 9 microtubules doublet, in between in the center, there is 2 single microtubules which are linked together with a intersinglet bridge. Intersinglet bridge. These single microtubules are known as singlet, and these singlets are joined together with the intersinglet bridge. So this is the 9+2 arrangement. That is why the cilia is given, the symmetry of the cilia is given a name of 9+2 arrangement. Now, in the starting of the video only I have discussed that these cilia are made up of special structures known as axonemes. So, that's why I have drew here. When we will cut the cilia, then we can observe the axoneme, which is known as the core of the cilia and flagella. Now comes the centriole. Centriole 9+0 arrangement. So why this 9+0 arrangement? So as we have already discussed in cilia, in the center of these microtubules, their singlets are joined together by the intersinglet bridge and giving that a structure of 9+2 arrangement. But here in the centriole we can see that in the center the microtubules are absent. In case instead of that microtubules in the center, there is a proximal proteinaceous hub is present, and the microtubules are arranged in a triplet form. 9 microtubules arranged in a triplet manner. 1, 2, 3, 4, 5, 6, 7, 8, 9. and the triplets are joined together with a link known as the intertriplet bridge. This bridge is known as intertriplet bridge. Just like in case of centriole, the link is known as interdoublet bridge. Similarly, in case of centriole, it is known as intertriplet bridge. Then, centrioles and cilia may look the same, but their function depends on where they are and what they connect to. The microtubules in the centrioles don't move, but when they form the cilia, they start moving. So let's simplify the difference. Centrioles are the organizers. Centrioles are the organizers and they help in the cell division and give rise to the cilia. And cilia are the movers. They beat rhythmically to move things along. Alright. Now that you have got the full picture of centrioles and cilia, next time you face this confusing topic, just remember centrioles are like the cell's directors, they organize the show. Cilia are like the performers, they move the audience or the fluids around. If this video helped clear up the confusion, hit that like button and don't forget to subscribe to Genaura Academy for more simple biology lessons. Thank you.